Hello, I am the Amoeba Brother, and welcome to your new summer job, DNA Replication. Now, here at DNA Replication, our job is to replicate DNA. We first start off with the original strand. We take the original strand, and at a region called the replication fork, our employer helicase goes and unwinds the DNA. However, the two sides are really attracted to each other, so we have a group of employees called the single-stranded binding proteins, also known as group SSB proteins for short, bind to the DNA strands to stop them from getting intimate together. While this is happening, our other employees are building the new complementary strand. However, the process is different for the two strands. Let's start off with the leading strand, which is a strand that's being made in the same direction as helicase in the replication fork. Before we get into that though, it's important to know how the different bases pair together. There are 4 bases and 2 sets of pairs. Bases adenine must go with thymine and guanine must go with cytosine. They form together with hydrogen bonds, which helicase breaks to unzip the DNA. Back to the leading strand, one of our employees' primase makes a small segment of RNA called the primer. After that, our star employee DNA polymerase 3 binds new nucleotides to the old nucleotides by building them off the primer. Heads up, DNA polymerase 3 and primase have a rocky relationship with each other, so I wouldn't sit next to them at break time. After this, DNA polymerase 3's younger brother, DNA polymerase 1, removes the primer and replaces it with DNA, while also correcting the mistakes his brother makes. They also have a pretty rocky relationship. Now to the other strand, also known as the lagging strand. This process is similar but more complicated. You see, the higher-ups maintain that our employees should only work from 3' to 5' due to keeping brand consistency. However, DNA strands are anti-parallel, so while primase and DNA polymerase 3 and 1 build the new strand the same way, they're doing it in the opposite direction that helicase is going. This causes them to lag behind the other strand, causing tension between the employees and the different strand. In order to keep up, they replicate the strands discontinuously, resulting in the strands being split in different fragments. We call these Okazaki fragments. While this is annoying, the higher-ups refuse to change it. Instead, they created a new position called ligase. After DNA polymerase 1 is done, ligase seals up the gaps between the Okazaki fragments, creating a continuous strand. At the end, we have 2 identical DNA molecules. Now, before we end this training video, we want to address the elephant in the room. Our company's politics have recently come into question. While we here at DNA Replication don't subscribe to any party, we identify as semi-conservative because each new strand we make has both an original and new strand. That'll be all for now. Congrats on getting hired and welcome to the team.